Good morning, viewer. Welcome to our up-to-be English version. Here's some breaking news. Research from PhD candidate Mohan Chen in the lab of Aldesi Hamster of the Development of Cell and Developmental Biology at the University of Michigan Medical School and their colleagues improves upon a popular experimental model, and in doing so reveals more of the inner working of a critical sphere during the formation of an embryo. The findings are published in the journal Nature Methods. Scientists study development in part to understand what can go wrong as the body structures are formed. The ultimate hope is to be able to prevent birth defects and identify causes of failed pregnancy. However, studying embryos in a laboratory is fraught with important ethical and technical considerations too. To alleviate these concerns, many researchers turn to simple 2D structures made up of stem cells called gastroloids. Gastroloids are grown, cultured in vitro in a dish and model some of the earliest moments of development without the potential to develop into a human being. Gastroloids recreate aspects of a process called gastrulation in experiments. During gastrulations, a so-called primitive streak of cells gives rise to three embryonic layers of the cells that ultimately make up the body plan. The ectoderm, outer layer of which gives rise to the skin, nervous systems and other outer structure. The mesoderm, middle layer, which becomes the heart, muscles, bones and other internal structure. And the endoderm, the inner layer that becomes the GI tract, lungs, liver and other organs. However, the in vitro culturing of gas typically only been sustained for up to 2 days. After that, the cells become organized and stop developing. Using different media and made several other improvements to the model, said hamster. And when we tried growing the cells for more than 2 days, it actually worked and did some really interesting things. That's the teams show that the stem cells of the developing mesoderm began to travel underneath the original single layers of cells, forming a multi-layered structure just as it would in a real embryo. This is very hard to visualize. We don't even know how it works in mice, said Armstrong. Yet, their extended model enabled them to see that the mesoderm stem cells migration is directed from the edge of the group of cells to the center. That means there is something. We don't yet know what telling them which way to go. We now have a setup in which we can figure out what is guiding their movement, said Armstrong. This is critically important for figuring out what goes wrong in certain cases, such as with the generation of congenital heart defect. The team also determined that the mesoderm cells in the gastroloid come in several different subtypes that express different genes, which they visualized using fluorescence. By looking at what gene the cell is expressing, you can kind of see which organ it will ultimately become, Hansard said. When this exclusive report, FTV English.